Okay, welcome back from spring break. The topics that we're going to be covering for the next several meetings, I think probably would have been more appropriate if we could have started talking about them before spring break. They have to do, and especially now with love in the time of cholera, I think that these topics would have been appropriate for you to prepare you for what's going on. We're going to be talking about sex and sex determination and control of sex determination. And we're going to start with several different examples, but the first of those examples will be most relevant to you all, which is humans. So sex determination in humans. So if we have an individual that is XY, versus an individual that's XX, what's the biological sex of the individual that's XY? Audience could answer if they wanted to.
Oh, male.
Male, that's right. Yeah. So this is male. And what about XX?
Female.
Female, okay. What about XO? So this is an individual that has one X chromosome and no second sex chromosome. Female, but sterile. So this is Turner syndrome. What about XXY? Male. Okay. So what is it then that determines biological sex in humans?
Chromosomes?
Well, chromosomes.
The absence or presence of the Y chromosome?
Yeah, the Y chromosome, right? So the Y chromosome determines the sex. Specifically, its presence leads to male determinant. And that has to do with genes that are on the Y chromosome, although there aren't very many that have been well characterized. And so basically having a Y chromosome means that that individual will be male biologically in humans. Okay?
Mm-hmm.
Now let's compare that to Drosophila, to fruit flies. So let's do the same thing. So XY in Drosophila, male or female?
Male.
Mm-hmm. Yep. XX.
Female.
Mm-hmm. XO.
Female.
Male. But sterile. Let's see. XY.
Female.
Actually, XXY is, I think that is female. Yeah, it's female. So, let's see if we have another example, XXY. So what is it that determines sex in Drosophila then?
Also the Y.
Well, does Y determine the sex though? So if Y determined the sex, then having a Y chromosome would ensure that you'd have a male.
The presence of a second X.
Yeah, the number of X's. So the ratio here, the ratio of X's to autosomes determines the sex. And if that's the case, then what is the purpose of Y? Does it have any role?
I mean, I guess it's just there to like, to balance out like that ratio, to like balance out the X.
Well, what do you see when Y is present?
Either one sex or the other.
Yeah, but there's also something else that happens here.
Oh, fertility.
Yeah, so Y is really for male fertility.
Okay.
So not having a Y means that you still can be male, just not sterile. You'll just be sterile. And having a Y doesn't make you male if you're a fruit fly. Okay? So what we're going to do next is we're going to try and figure out what's the basis for this pattern in Drosophila. Okay? And the reason that we're bringing up this particular example now is where we ended before spring break was talking about alternative splicing. And what we're going to find out is that this particular method of control involves alternative splicing patterns. So, let's see, where's this? So in Drosophila, we said that the ratio of X chromosomes to autosomes is what makes the determination. So if we have a high ratio, we're going to get one set of circumstances taking place, and we're going to get a different set of circumstances if we have a low ratio. And by high, essentially what we mean is 2 X's versus only 1 X. Okay? So if there's a high ratio of X to autosome, and those of you who are considering your next wiki, which will still be due even though we're working remotely, in April, coming up, we're not going to talk about all the details of this, but we'll talk about some of the details that lead to the final outcome. So having a high X to autosome ratio is going to change splicing patterns. And one of the things that's going to be spliced differently is the RNA that gives you the messenger RNA for a protein called SXL. Okay? So this gives you alternative splicing of the SXL mRNA. And the result is that you get a functional SXL. The other mechanism of splicing that could take place for this protein, for the messenger RNA for this protein, would give you a messenger RNA that has a premature stop codon in it. So the result would be that no SXL functional protein would be made. And that will be what we talk about on the other side of this picture. So for this picture, because of the high ratio of X to autosome, we get alternative splicing that gives us a messenger RNA that provides a functional SXL. Now, SXL is itself an alternative splicing factor. And the result is it's going to change the splicing pattern of the RNA for another protein called TRA. And that'll give us a functional TRA protein. And because that protein has been made, it's going to do alternative splicing of a second splicing factor component. Called TRA2. And once you have both of these proteins produced together, that gives us an alternative splicing factor of a category of splicing factors called SR proteins. And that's, the S stands for serine, and the R stands for arginine. And so these proteins, these alternative splicing factors, tend to have serine and arginine as part of their functionality. But the thing to really remember and take home is that together, TRA and TRA2 form yet another alternative splicing factor that act on the RNA for another protein called DXL, DSX. And DSX is a transcription factor which allows for expression of female-specific genes only. So if you follow this pattern of alternative splicing, when the ratio of X to A is high, you get the production of alternative splicing factors that in a series of events splice the RNAs for other alternative splicing factors that ultimately lead to alternative splicing of the RNA for a transcription factor, DSX, and that specifically allows for female-specific genes to be expressed. Okay? Any questions? Yes?
That seems like such a complicated pathway to finally like get to that one specific thing. Like, what if something goes wrong?
Yeah. Well, things can go wrong. Some of these proteins, the reason they have those names is what happens when they go wrong. So as we've talked about before, the way geneticists do things is you make mutants. And so when you make mutants, you see what happens in terms of the change of phenotype. So SXL, I think, stands for sexless. So if you don't make this protein at all, in some circumstances, it affects the fly, so it doesn't really have a biological sex. And then mutations affecting DSX, I think there's some mutations that can lead to what's called double sex, so they have characteristics of both. So things can go wrong. But on the other hand, if you think about it, the control of biological sex is pretty important for a species to be able to persist. So you'd want to make sure that everything was done properly. All right, so what happens then if the ratio of X to A is low? Well, if the ratio of X to A is low, you get default splicing. And the result is you get no SXL made. And if you get no SXL made, then what else are you not going to have?
Eventually, DSX.
Right, so you've already cut to the chase, but as we do it step by step, because you have no SXL, you get no
TRA.
That's right, so then there's no TRA, because we didn't alternatively splice that. Because there's no TRA, then we don't get
TRA2.
That's right, so then you get no TRA2. And without TRA and TRA2 to give you that splicing factor, you get the default splicing DSX. And the result is that you get a DSX transcription factor that is male specific. And the female genes then don't get expressed, or female specific genes don't get expressed. Okay, any questions about that? Okay, all right, so this is one example of sex determination, and again, it's interesting to compare how the determination is made here versus what we saw with humans in terms of the sex chromosomes involved. What we're going to move on to now is an even more interesting example, because I know that many of you are interested in fungi, like I am, or not. But we're going to talk about how sex is determined in fungi. Okay? So I'm going to erase this, which everybody now has permanently in their memory bank. So in terms of fungi, the first fungi we're going to talk about that are involved in this process are Saccharomyces cerevisiae. Or yeast. These are budding yeast. So they reproduce by cells that look like this. And when the cell is going to reproduce, you get this kind of a budding pattern, and then eventually you have the original cell and the daughter cell, which separate after budding. And this process occurs when there is sufficient nitrogen and fermentable carbon. Now, what's interesting about yeast is that they have both haploid cells and diploid cells. And under these kinds of conditions, either type of cell can reproduce by budding. Okay? Now, there's something else that happens with yeast, and that is these haploid cells can come in two flavors, if you want to think about it that way. There are A cells and alpha cells that have what we call different mating types. And the A cells, they produce a pheromone that is produced from a gene and then processed into a final smaller peptide. So the active pheromone is 12 amino acids, and the alpha pheromone is 13 amino acids. And the A pheromone actually has some chemical modifications that are added to that small peptide after it gets produced. One of those is that a lipid group farnesyl group is added. And there's methylation of the carboxy end of the protein. So you also get carboxy methylation. And that's for the A pheromone. So each of these mating types has their own pheromone, and then let's say that these are haploid cells. So this is A, and no, let's not do it that way. Okay, so here's your A cell, and see if you can see this red, I hope. Yeah, that's better. Here's your alpha cell. So the alpha cell secretes a pheromone, and the A cell has a receptor on its surface for the alpha pheromone. And the A cell, similarly, secretes its pheromone, and on the surface of the alpha cell, there is a receptor specific for that. Okay? And that's how those haploid cells communicate. And what happens is, once that occurs, there's a series of signal transduction steps that occur inside the cell that we'll talk about later. But the main thing that happens is they move towards each other and they produce a structure that is part of their fusion process that winds up being something that looks like this. And this is referred to as a shmoo. And initially, the nuclei are separate, but eventually the nuclei fuse, and the result is that you get a diploid cell. During this process of fusion, though, you get a rest in G1, and the cells fuse, and eventually you get the production of a diploid cell. Which we'll refer to as an A alpha diploid. Now, diploid cells can reproduce by budding. But there's something else that they can do that haploid cells can't do, and that is they can also, under certain conditions, they can also sporulate to undergo meiosis and produce haploids again. Okay, so this is kind of their mechanism of sexual reproduction. Any questions so far? All right, now this is where it gets even more interesting, because it turns out that these haploid cells can switch mating type. So haploid cells switch their mating type, and it depends on a separate genetic locus than the one that determines whether they're A or alpha. So there's one genetic locus involved in being A or alpha, and then there's a separate genetic locus that determines whether or not they can switch. Okay? So this depends on having the dominant HO allele, which encodes an endonuclease leads to switching of mating type every other generation. So that's pretty frequent. And just to emphasize what that means is if a mother cell produces a daughter cell, the daughter cell can't switch, but her daughters can switch. Okay. And then there's a separate allele to that, which is recessive. In which case, switching is rare. On the order of about 1 in 10 to the 6th cells. So any questions so far? Okay, so what we're going to do is kind of make a table that summarizes what we just said, and then we'll start looking at some of the details of how this actually works. So let's see, I'm going to erase this. Okay, so what we're going to try and see is how much you all remember from what we just talked about. So first of all, if you have a mat A cell, so this is a meeting type A, do they make pheromone? So mat A cell that's meeting type A, do they make a pheromone?
Yes.
Yes, I guess, that's right, wasn't a trick question. So, and if so, what kind of pheromone do they make?
The
12 amino
12.
acid one.
Right, so they make the A pheromone. So
The
these guys
pharmacy.
make A pheromone. And what about the mad alpha?
It also makes pheromone,
And which type of pheromone would it be?
the alpha pheromone.
That's right. And what about receptor? So for the mad A, do they make a receptor?
Yes,
And it would be the receptor for,
alpha.
that's right, so receptor for alpha. And what about the mad alpha?
Receptor for A.
Right there. All right, can mat A mate?
Yes.
Yes.
Yes. Can mat alpha mate?
Yes.
Yes. Okay. Now, can mat A sporulate?
No.
not unless it's together, right?
Right, so in other words, so a haploid cell can't sporulate, which makes sense because sporulation, part of that is meiosis, and a haploid couldn't undergo meiosis.
Mhm.
So Mat A cells cannot sporulate, and what about Mat alpha?
No.
That's correct. Now the question is, what happens with the diploids? So just as a reminder, these guys are diploid. Okay, so first of all, can a diploid sporulate?
Yes.
Yes. Can a diploid mate?
No.
No. And the reason for that is, does a diploid make a pheromone?
No.
No. And does a diploid make the receptor for a pheromone?
No.
No. Okay? So this is kind of the overview in terms of the rules of how these are supposed to behave. And what we're going to do is, from this set of information, we're going to move on to discuss sort of the molecular genetic mechanisms that control this pattern, or this set of patterns that we see. Okay? All right, so you can